it's just a lot of organization of content. It's a content-heavy class. You have to organize it in your brain, okay? It helps you to find the answer when you get a test question, whether it's verbal on the lab quiz or written on lecture exam 1. You want to find the answer. You don't want to be like, where do some in there somewhere, it's doing this on me. You want to find it. And it takes discipline to organize content in your brain. Okay, it doesn't just happen, it takes intention. So I teach it intentionally to be organized, to help you, to show you how to do it, how to organize content. Are we ready? Look on page 5, it's over there on the right screen. But turn to page 5. We are covering this in lab today. We will be able to see some nuclei. That's an organelle we saw today in lecture. We're going to see cells, but we're really talking about the 4 tissue types. That's why I put the number 4 in parentheses after tissues. This is what we're studying today in lab. Next week, we're going to already go up to here. This is our only lab down here. Are we clear? There are 4 types of tissues, and it's listed in order. The way you start by being disciplined with content, keep it in the F in order. First is epithelial, boom. Second is connective, boom. Third is muscle tissue, boom. Fourth is nervous tissue. Next week we're going to add the integumentary system. But today it's just the 4 tissue types. 1, 2, 3, and 4. 1, 2, 3, and 4. Keep it in order. Okay? Now, right away I'm going to show you here how this is your best friend. You'll see about it in just a few minutes. There are actually 8 epithelial tissues. 8. Your best friend only has 6. One of them that's not listed on here, stratified columnar, looks very similar to this last one. Pseudostratified ciliated columnar epithelium. They look very similar. Guess what? You don't have to worry about distinguishing the two. Because if it looks like stratified columnar, it can't be stratified columnar because stratified columnar is not one of the 6 listed here. If it looks like stratified columnar, it's fake stratified. Pseudostratified ciliated columnar epithelium. Are you getting how this is your best friend? But only if you use it. If you don't use it to help you critically think, you have wasted a gift that could help you pass this class. So let's go through it. First of all, epithelial tissue we're going to do first. Why? By keeping it in order. It doesn't matter if it's 1, 2, 3, and 4. You can do A, B, C, or D. Doesn't matter. But keep it in order. Epithelial first, connective tissue second, muscle tissue third, nervous tissue fourth. Are we clear on what the 4 tissue types are? Everything I'm going to teach you this semester, if you look at all the cabinets, so this is room to share with zoology. Of those models back there, we have more anatomy models in here. We have anatomy models up on the top, and wall charts. All of it is made up of 4 tissue types. Everybody got it? This is all microscopic. That's what, by the way, if you want to know what histology means, it means microscopic anatomy. Okay? So let's start with epithelial tissue. It says there on the very top of page 5, line or cover. Epithelial tissue is a lining tissue. Okay? And so there's going to be some things we're going to see in these cells that line stuff. It could either line stuff on the outside, like my epidermis here is epithelial tissue, or can line stuff on the inside, like lining the inside of your digestive tube. Any tube is lined on the inside with epithelial tissue. Are we clear? That lining tissue can be classified by how many cell layers thick it is. You guys are recording this, right? If it's only one cell layer thick, we use the word simple. If it's 2 or more layers thick, we use the word stratified, which means layered. And then we will look at the shapes of the cells on the top. And if the shape of the cells on top is flat or wider than it is long, we use the word squamous. If they are about as tall as they are wide, we use the word cuboidal. They're actually not cube-shaped, they're six-sided, hexagonal. Or if they're really tall, taller than they are wide, like a column, we use the word columnar. For this PowerPoint presentation, the number of cell layers thick will be in yellow. The shapes of the cells in the free surface will be purple. Everybody got it? Now look on the top of page 5. Under epithelial tissues, do you see some of those words? Do you see simple and stratified? Squamous, cuboidal, columnar. Now you know what they mean. Do you see why I covered this first? Okay, there is one exception that does not fit, and there's only one exception that does not fit, and that is transitional epithelium. You will not see yellow or purple words with transitional epithelium. This, at the very bottom, is a note of visual recognition. These are the types of notes I would make in an anatomy lab. Epithelial tissue, tissue type number 1, does not have blood vessels. That's what avascular means. Okay? You'll never see blood vessels in epithelial tissue. And since it is lining stuff, either on the inside or outside, it's got to be stuck to something, and it's stuck to a basement membrane. I'll show you what that looks like in a second. It is stuck to a basement membrane on one side, and on the other side is nothing, like this, because this is lining the outside of my body. It's stuck on the inside to the basement membrane, and out here there's nothing. We call this nothing the free surface. Now, if that's when you're lining stuff on the outside, you'll see the free surface. If you're lining stuff on the inside, like the inside of a tube, it's the hole in the tube is the free surface. Are we clear? So, this is a slide of visual recognition. What you're going to see is just these tissues don't look like this in your body. They're all stained. And they're stained to show nuclei. So you're going to see a bunch of purple balls or oblong. They doesn't have to be spherical, it could be oblong. You're going to see a bunch of stained nuclei. They're stained purple. And on one side, that stain comes to an abrupt end. But you can tell it's still in tissue. Tissue on one side, but all the stained nuclei come to an abrupt end. That's the basement membrane. That's on one side. On the other side is nothing. That's the free surface. And the epithelial tissue, look at me people, is in between. The epithelial tissue is between the basement membrane and the free surface. You're not worried about the distinctions of tissues 2, 3, and 4. You see that, you're in 1. Epithelial tissue. Are we clear? And again, it could be lining the inside of a tube. This is a kidney tubule. And this purple line is the basement membrane. There's all the stained nuclei all in a row. But the hole in the middle of the tubule, referred to as the lumen, that's the free surface. Yeah, if you didn't get the lab PowerPoint, this is the last one I'm printing, or no, I have PowerPoints for next week's lab for you since you're a week late. So that's the last one. So if you don't have one, you should have picked one up here. But notice, look at this. This is too sweet. I only had to move one arrow. Everybody see that? Okay, how many cell layers thick is that? From the basement membrane to the free surface. 1. What's the yellow word? Go back to that slide. 1 cell layer thick is what word? Simple. These cells are about as wide as they are tall. Now don't get your underwear in a knot just because this one is a little bit taller than wide and this one's a little wider than tall. You take the average, they're about as tall as they are wide. Everybody see that? So what's the purple word? Cuboidal. This is simple cuboidal epithelium. You see how those yellow and purple words work? Hope so. This won't take me too long to get through this if it don't. Okay? Any questions? Yes?
What is written in yellow? Like the yellow, that part. Okay, I see it now.
Okay. Everybody engaged? Again, if you get sleepy, you will disengage and have no idea what the yellow word is. Stay engaged. Everybody got that now, that this was simple cuboidal epithelium? By the way, if you look on the top of page 4, simple cuboidal will only get you a half point. For the lab quiz, it's you have to add either epithelium or epithelial tissue after those 2 words. Everybody got it? If you just write simple cuboidal, that's a half point. All right, let's go through the 6. Here we go. 1 cell layer thick, and they're all flat. The rest of this stuff, I use this from the lecture PowerPoint, because these take a long time to make. You're just making notes of visual recognition in this room. Are we clear? Don't worry about allows for diffusion and exchange. Don't worry, that's for lecture. This is, what does it look like? Well, you look at this and you think, yeah, that looks like more than 1 layer. No, it is just 1 layer. It's just that if you look up from the top of this, it looks like that. Does that help explain it? Just looking at this on top looks like that. I would never use this for the lab quiz. You'll have to see from the side that it's 1 cell layer thick. You'll see the basement membrane and the free surface. When you look on epithelial tissue from on top, you can't see the free surface or the basement membrane. Everything, by the way, in 2 weeks, the lab quiz, everything, every picture, in every version of that lab quiz has the things on the screen that's coming out of my mouth right now. Everything. If you let what's coming out of my mouth go past your ears, that's on you. Okay? So yeah, you might want to record my lectures for lab too. Stratified squamous. This I want to show you, and I want to mention a couple things. Number 1, You don't ever need to write for the lab quiz, keratinized or non-keratinized, but they look different, so I'm going to explain that right now. Epidermis is keratinized. And the way you can tell it's keratinized is all those cell layers at the free surface are dead. And you can't see nuclei in them. This is just the corpses of epithelial cells. That's all that you see. Everybody got it? But when you can see nuclei all the way up to the free surface like this is not epidermis. This is non-keratinized. And you can tell because you can see nuclei all the way to the free surface. Also, the purple word. You might want to write this on that slide. The purple word is all about the shapes of the cells at the free surface. Write it down next to this box. The shapes underneath don't count. The purple word depends on the shapes of the cells on top at the free surface. And we see that here in the slide. Because down here at the basement membrane, and that's where all the stain comes to an abrupt end, yes, it's not really high quality, but you can see some columnar, definitely cuboidal. None of that matters. When you get to the free surface, they're all flat, so the purple word is squamous. Everybody understand what it means by the shapes of the cell at the free surface? We've already seen this once. Again, they're usually six-sided in shape. You look at this and you think, God, that looks stratified. No. This is one simple cuboidal epithelial kidney tubule. It's stacked on another one. They're separate, simple cuboidal epithelial tubules. Everybody got that? They're actually sharing a basement membrane in between it. In the kidney, sometimes you'll see a capillary in the basement membrane, and that's a red blood cell, the capillary. But we already seen this. The difference is, everybody look at me, this kidney tubule is running this way. The microtome does that cut. Everybody see that? What we saw before was here. Except this kidney tubule is going like this. Everybody got the difference? This is called a cross section. This is called a longitudinal section. Everybody got it? Is this helpful? Because I'm busting my ass here. If this is not helpful, I can take it easy and make you teach yourselves.
So we're just supposed to know, because like you covered it, that this is simple and it's not stratified?
Simple cuboidal epithelium. I'm going to ask you, what is the name of the tissue? Simple cuboidal epithelial tissue, or simple cuboidal epithelium. It could be in this view, or it could be in that view. It's the same answer. Does that answer your question?
Yeah.
Transitional again, you don't see yellow or purple words. You see a slide that looks like this, and you'll think, it's stratified something, and it is stratified. It's actually 3 to 8 cell layers thick. Okay, so that's a yellow word. Let's go to the purple word. And then you see squamous, cuboidal, columnar. You have multiple cell shapes at the free surface. That should tell you there is no purple word. This is the one epithelial tissue that does not fit. There is no purple word. This is transitional epithelium. That is critical thinking. Process of elimination that an engaged mind can do. A disengaged mind can never do critical thinking. You're grasping for straws. Stratified, yes. 3 to 8 layers. So you're thinking, oh, I'm going to give it this yellow word, stratified. Let's look for the shape of the cell at the free surface. Squamous, cuboidal, columnar. There's no purple word. This is the one that doesn't fit, transitional epithelium. This box on the bottom, isn't that a note of visual recognition? Probably something you should highlight. Simple columnar epithelium is the predominant tissue lining of your guts. And we talked about this I mentioned this in lecture today, but only Monday Lab has heard this before since we didn't have Lab this last week. But the microvilli, this border at the top is very diffuse because of microvilli. That's where we see it because your guts are absorbing a lot of nutrients. Here's what you want to write next to simple columnar epithelium. Yes, the cells are taller than they are wide, but there's only enough room for the nuclei to be in a row. All the nuclei will be in a nice little row. That's how you know it's simple columnar. Are there pictures of simple columnar where they're not in a row? Yes. Guess what? I won't use it. All the images that will be on the lab quiz have the things that are coming out of my mouth right now. You just have to visually recognize.
What about the ones below it?
This is actually in the lamina propria. This is connective tissue down here. So it's only one cell layer thick. This right here is the basement membrane. This is tissue type number 1, and it's one cell layer thick, simple columnar epithelium. This is loose connective tissue. Okay. So I don't know what this connective tissue looks like. Well, you will in a bit. But all you have to know is see that these are, all you have to do is use your best friend. I'm going to say, what is the tissue between this line and this line? You're going to see the stained nuclei in a row really close together when they abruptly stop. You're going to see a free surface, which is the lumen of the guts. You're going to know this is lining the guts or it's lining something. There's a free surface. That's the white area in the middle. You're going to say that's tissue type number 1. How many cell layers thick is it? 1. Are they taller than they are wide? Yes. Columnar. Are the nuclei in a row? Yes. Simple columnar epithelium. Move on to the next one. You know that's the right answer. Everybody got it? You can't do that if everything I'm talking about today is diffuse and disorganized in your head. If it is organized and in order, you can reduce these images to a game of 20 questions. Have you guys ever played 20 questions? Because not too many people do anymore. We do that on long car rides on vacation. But now nobody interacts because they're doing this. But if you play 20 questions, I'll go over some 20 questions a little later and show you how to do this. It only works if the content is organized and in order in your brain. Okay. Last one is pseudostratified ciliated columnar epithelium. It is a mouthful. I would write it like this. E before U, pseudostratified ciliated columnar epithelium. I just described you the most common missed half point of the lab quiz. E before U, pseudostratified. It's nice because it rhymes. E before U, pseudostratified. This predominant tissue lining of the respiratory tract. And the organelle we talked about in lecture today is this black arrows on the cilia. There's a mucus layer on top secreted by these goblet cells. Talks about in here somewhere. on another slide. But it secretes a layer of mucus, and the cilia here is beating that mucus layer to your throat. In your nose, it's beating backwards, and in your bronchial tree, it's beating upwards. So in case you inhale something that don't belong, it lands on the mucus layer and you can get rid of it. Everybody got it? All right, so for tissue type number 1, here's all 8 on this slide. Put an X through this one, and an X through this one. You only have to know these 6 and you're best friend. And in histological slides, this looks very similar to this. Because let's look at that again. This is an image that's in your lab quiz. You're going to say, well, that's stratified. Oh, but at the free surface, these are definitely taller than they are wide. This is definitely stratified columnar. Except look on the top of page 5. Do you see stratified columnar? It can't be stratified columnar. It's not your best friend. If it looks like stratified columnar, it's pseudostratified ciliated columnar epithelium. That is your best friend, if you use it. Everybody got it? Yes?
Which ones did you want us to cross out again?
What's that?
Which ones did you want us to cross out again?
Oh, on that picture that has all 8,
Yes.
these 2 in the middle. Those are the 2 that are not in your best friend. Everybody got it? You got 6. What are you looking for tissue type number 1? This is cool. I can hear my voice is coming back. What is it? It's actually 2 things. Basement membrane and a free surface. If you see a basement membrane and a free surface, you are tissue type number 1. Forget the other 3. Are we clear? What does a basement membrane look like? It's where all the stained nuclei come to an abrupt line in the tissues. Again, I'm saying it. If you let these words go past your ears, where it does not help you in 2 weeks, that is on you. I said it. You just didn't hear it. I said it 4 times and you didn't hear it. What are you looking for tissue type number 1?
Basement membrane.
Basement membrane and a free surface. What does the basement membrane look like?
Abrupt line of nucleus.
Where the stained nuclei come to an abrupt line in tissue. Like we saw it in this last slide. Look at all these stained nuclei. It comes to an abrupt end in the tissue. This is the basement membrane. That's the free surface. Tissue type number 1 is in between. Everybody got it? Tissue type number 2. I'm going to try to get through this before we break again. This one is the hardest out of the 4 tissue types. I'll just tell you right now, this is the most difficult. But the thing that will be key for tissue type number 2, connective tissue, is that these are cells, here's the cells here, living in non-living stuff we call matrix. Cells living in non-living matrix. Because the cells are separated by matrix, this is what gives you the key that you're in tissue type number 2. The cells are separated by matrix. They are not close together. Put an asterisk next to them because there's one exception. But that one exception is easy to visually recognize. So don't worry about it. The rest of this, again, these are cells and these are the matrix that's separating cells. But I left this in because even though you don't need to know the particulars of those, after the first diamond here, write the word pink. Collagen, for our purposes, where it's relevant, is going to be stained pink. And next to the second diamond, write the word black. Elastic fibers are much thinner, and they will be stained black, where it's relevant. Are we ready? You ready to go on to all these? All right, there's actually 6 connective tissue propers in your best friend. I only have half of them. Don't need to worry about the other half. First, and the correct spelling for the full point is in the top row of this table. First one is either loose connective tissue or aurelian connective tissue. Which one is easier to spell? Loose, just call it loose connective tissue, okay? I have students trying to impress me by writing aurelian or loose connective tissue. And they misspell aurelian. They spell loose connective tissue correct, still lost a half point because they misspelled aurelian. So don't even bother writing it, just write loose connective tissue. Or means you have a choice. Choose the easy one. That is also critical thinking. What does loose connective tissue look like? It looks like you've taken all those different cells, fibroblasts, macrophages, mast cells, and gone like this, splat. And you took pink collagen and black elastic fibers and go splat. That's loose connective tissue. It is a mishmash of cells mixed with a mishmash of fibers. Can you see the pink in here? They're a little bit thicker. That's collagen. Do you see these really thin black? That's elastic. Where you put the asterisk for cells, so cells are not close together, except asterisk. And that asterisk goes on adipose. This is the only connective tissue where the cells are touching. But fortunately for you, adipose is easily identified because they look like extremely thin walled bubbles. The reason why is adipose sites, once they start to store fat or oil, same thing, they stop dividing and they stop doing just about anything else except storing and then releasing fat. And fat, or the oil, is not visible in microscope slides. They don't stain. So these are extremely thin walled bubbles. And the wall of the bubble looks as thick as those purple lines. What it says there is that, to an untrained eye, some people might think this is a simple cuboidal epithelial tubule. And cross-section in the kidney. This is what you're going to school for, so that you can learn what the difference between things that look very similar, so that you can have trained eyes. Right now it's untrained. And here, simple squamous the wall is actually thicker. That purple line, and not this purple line. Does this picture here help explain what's on the bottom right of the slide? They look the same to the untrained eye. No, one's thicker. That's the simple cuboidal epithelial tubule. I'll show you a picture of it later. But these are extremely thin-walled in appearance. The actual wall, cell for an adipocyte is the whole circle. The whole circle is the thickness of the cell. A squamous cell, that's the thickness of the cell. But an adipocyte, the fat doesn't stain, so it looks clear. Does everybody understand what I just said? Yes?
So the lumen is a hole.
Lumen is the hole in the tubule.
And so then, and the other one, since it looks clear, they look similar?
Yeah, it's not a hole, it's full of fat.
It's full of fat. No.
Hey, guess what? Good news. The amount of fat cells you have, is as high as it'll ever be right now. You'll never get any more fat cells. Isn't that good news? No, the bad news is there's no limit to how big these cells can get. It's always bad news, no, it's never all just good news. Which is what the sum of we talked about in lecture day, about having superior opinions. Here's an inferior opinion. You want to get skinny, just do liposuction. That is a freaking inferior opinion. If you do liposuction, you have just removed adipocytes. You have got to stop eating, and you've got to start exercising if you want liposuction. Why? That's the thing that you needed to get rid of that fat in the first place. If you do it surgically and don't change those things, the fat in other parts of your body are going to make up the difference. And you're going to have these huge water wings of fat arms wherever you didn't have the liposuction. Everybody got it? This type of science will help you to make better decisions, not just for yourself, but for your family. You're not educated. You think I can just fix this with surgery. Well, no. You're going to really have to change once you have the surgery, and if you really changed, you wouldn't need the surgery. Does that make sense? All right, and the third and last connective tissue proper is dense regular connective tissue. What you want to write next to this one, and again, you need the 4 words in the box for the full point. Dominated by parallel lines. They don't have to be straight, they can actually be wavy depending on how it was cut, but they're parallel. Dominated by parallel lines with very few nuclei. Because these lines are not cells, they're collagen, that's why that's pink. We're going to look a little bit later at smooth muscle. Same thing, parallel lines. That's what it's saying in the bottom. Notes of visual recognition, people. Smooth muscle, dominated by parallel lines, but every line is a cell. A spindle, thin shaped cell. That's why smooth muscle is loaded with nuclei. Here, the nuclei fibroblasts are staying in these little slit-like black lines. But notice how those slit-like black lines are separated by stuff. Connective tissue, tissue type number 2. Except for adipose, the cells are separated by stuff, matrix. Everybody got it? I would write parallel lines, very few nuclei. Then we have supporting connective tissue, that's cartilage and bone. Cartilage and bone have lacunae. What are lacunae? Lacunae are the spaces in the matrix that the cells live in. Now let's just jump to bone so I can get bone over with really fast. Here you can see, this is the singular spelling, lacuna. You put an E at the end, and that's the plural spelling, lacuni. Everybody see that? Singular, lacuna. In bone, if I show you a slide of bone, it's going to be compact bone. And in compact bone, we have these complex structures called osteons. What osteons look like is a cut tree trunk. It looks like it even has tree rings. So if it has lacunae, but it looks like a cut tree trunk, that's bone. And you only need 4 letters for the full point. Everyone know how to spell bone? That's it. That's all you need. Any questions? This is how I can tell when students are disengaged. I've had lab quizzes come in, and next to this line where this was asked, it says cut tree trunk. That's a disengaged mind. Cut tree trunks are way bigger than this, people. Osteons are microscopic. But you just write bone. You don't have to say osteon, just write bone. But if you have lacunae not in a cut tree trunk, it's got to be cartilage. And what lacunae look like in cartilage, it looks like a light halo around the cell. In fact, look at that guy. It looks like a little eyeball looking at you. Same thing here. Same thing here, 3 right in a row. Now, sometimes you'll actually see the cells touching. So it's not 100% that the cells are not touching. It's mostly the cells are not touching. When you have a situation like this, you know why the cells are touching? These cells just divided. And they're about ready to migrate apart and be separated by stuff. But right now, they're stuck together. Does this make sense? Okay. So when we get to cartilage, you're looking at cells separated by stuff. You know you're in connective tissue, tissue type number 2. You're going to see halos around the cells, lacunae, not in the cut tree trunk. And you're finally going to start thinking about fibers. If you see black disorganized fibers, you know black is elastic fibers. This is elastic cartilage. 2 words for the full point, elastic cartilage. If you see lacunae, cells separated by stuff, not in the cut tree trunk, and you see roughly parallel, it doesn't have to be this exactly parallel, but roughly parallel pink fibers, that's 1 word, fibrocartilage. 1 word. It's not fibrocartilage, 2 words, it's fibrocartilage. Everybody got it? Now I would never use this slide on the lab quiz because you can't see the lacunae. All versions of each one of my lab quizzes has things in it that are fair, because I teach defensively, and that means I teach to minimize student bitching. And there are some slides that are bad that has legitimate student complaints attached to it. None of my slides have that. So don't even try to bitch at it. This one would be about it. I won't use it. But this is what you're going to see. You're going to see lacunae, halos around the cells, not in a cut tree trunk, and it looks like all those cells are in jello. You can't see the fibers. There is actually collagen fibers there, it's just so sparse you can't see them. Are we clear? So it's not visible, there are fibers, you just can't see them. Black disorganized fibers, roughly parallel pink fibers, jello. And hyaline cartilage is 2 words for the full point, hyaline cartilage. Are we clear? Okay, I'm going to come back to this when I talk about 20 questions. But the last connective tissue is blood. And blood is going to be dominated by pink to purple circles without nuclei. And this is what I was asking in lecture today. This is an example of a cell that does not have nuclei in it. They did when they were in red bone marrow, but before it got spit out, it spit out the nucleus. The few cells that you do see with nuclei, those are white blood cells. But all these other ones that dominates the slide is red blood cells. The little speckles are platelets, by the way. Okay? But you see that, that's blood. 5 letters for the full point, just blood. Any questions? Any questions on how to write connective tissues to get the full point? Because I went through all of them. Unless that went past your ears. Okay, before we take a break, I want to talk about this. 20 questions. So when my kids were young, we didn't have this. So we went on a long trip to Grand Canyon or wherever, Lake Tahoe. We played 20 questions. I'm thinking of something. You have 20 questions to pare it down to what it is. And Bryce, who's in charge of the STEM tutors over there in the STEM center, he hates it when I tell this story, but that's okay. But he sucked at 20 questions. So, okay, I'm thinking about something. Bryce will go, is it our neighbor Zach? No, and you just lost the question. You got 19 left. And that didn't even help you. How do you play 20 questions? You start out with the general questions first. And you get gradually more specific. Is it animal, vegetable, mineral, or manufactured? It's animal. Okay. Is it an animal in a zoo? No, that's 2 questions. Is it an animal like a pet? No, that's 3 questions. Is it a human being animal? Yes, that's 4 questions. Is it a famous human being? No, that's 5 questions. Is it a human being we know? Yes, that's 6 questions. Is it someone in our family? No, that's 7 questions. But it's someone we personally know? Yes, that's 8 questions. Is it someone that lives near to us? Yes, that's 9 questions. Is it our neighbor Zach? Yes. Do you see how you go from general to specific? And you can do that more quickly than just starting out, is it our neighbor Zach? First, get which of the 4 tissue types is it. Do that first. Now we already know that we're looking for basal membrane and a free surface for tissue type number 1. We're looking at cells that are not close together with the exception of adipose for tissue type number 2. 3, muscle tissue with stacked tubes. 4, nervous tissue has no tissues. So the question that's going to come out of my mouth is not going to be, what tissue is this? That's the quickest, easiest way to start your 20 questions. Basal membrane and a free surface. Epithelial tissue. What's connective tissue?
Living cells and non-living.
What's that?
Connective tissues, living cells and non-living matrix.
Cells are not close together with the exception of adipose. 3, stacked tubes. 4, there are no tissues. Now you can get close really fast. If you do see a basal membrane and a free surface, boom, you're thinking how many cell layers thick and what is the shape of the cell at the free surface. Those are questions you will not ask for tissue type number 2. Are you following me? But say we are tissue type number 2. Let's say it's the one in the middle there. I don't see basal membrane and a free surface, but I do see lacunae, a whole bunch of lacunae. It's not in a cut tree trunk. I know I'm in cartilage. The last questions I'm going to ask is about the fibers. I see roughly parallel pink lines. Fibrocartilage, and I'm not guessing. I did 20 questions really fast, didn't I? You don't even have to have seen the picture before. A brand spanking new picture. You apply 20 questions. Boom! You get to the answer without guessing. Who wants to do that? Who wants to achieve number one on their card? You don't get there overnight. You start by doing this. Then you can do this. You understand what I'm saying? All right, let's go ahead and take a break when that clock says 2:45. I'll finish the last